All right. Oh, okay. There. Hello. Can anyone hear me? Oh, okay, we're good. Awesome.
Yay!
Yay, there we go. Just wanted to check. Okay. Hello, everyone. Welcome to the Cancer 101 panel. This is where we discuss how your donations save lives. And we really wanted to do this because all the things you're doing here have a real tangible impact on cancer research. And we just want to give you all just a rundown of what that looks like, because it's very easy for all of us to be like, we gave $10, $15, $20 to PCF, and then we never hear anything ever again. So this will be a peek behind the scenes, so to speak, of where exactly those research dollars goes towards and what good it's doing to the world. So thank you for showing up. This is just a summary of what you saw on the website, or summary here. So we'll go over the main big topics of this panel in a couple slides here. But before we do so, let's introduce ourselves. So I have Satan with me, if you'd like to introduce yourself.
Yeah. Hi, my name is Satan herself, and you might be like, what is she doing here? Well, actually, I did spend a year of my life as a cancer researcher in a cancer biology lab, and I have a neuroscience degree. And I have a specialty background in molecular and cellular biology with some genetics research, some genetics background as well. So that's why I'm here. I'm also a Games and Quick host, runner, staff member, all sorts of stuff, and I'm excited to talk to y'all. Yeah.
Cool. Yep, and hi, my name is Caritas, KRT, but you can call me Curry for short. And so my background is I have a PhD in genetics and genomics. I do research on rare human genetic disorders. I haven't published directly in cancer, but I've worked on cancer data sets. But I've also worked on other genetic diseases like sickle cell disease, glaucoma, neural tube defects, and a few others. But yeah, I'm also here because I know a fair bit about the genomics of cancer and cancer progression, and I just wanted, and also about how research happens in terms of getting grants and translating dollars into information, into cures and treatments and screenings and preventions that can be used in cancer. So yeah, glad to be able to share all that with you today. So here's a very basic outline. We have 4 big sections for you. First is just Cancer Biology 101. What is cancer? How to detect cancer? How does cancer progress? Second, cancer/research funding. How does the money work? Where does it go? Third is a project specifically by PCF to give you an outline of, again, where exactly your donation dollars are going towards. And then the fourth one is just the latest advancements in cancer screenings and technologies, just to give you a glimpse of how fast and how much we know about cancer, how rapidly that's progressing. It's a truly exciting field. So
Yeah, and then at the end, we'll have time for questions, and we do have a special guest for our questions. This is Jody. She is the CEO of the Prevent Cancer Foundation. So she will be here to answer your questions as well at the end if you have any specific ones for her, as well as, of course, ones for us as well.
Mm-hmm. So one more thing before we get started, just some disclosures and whatnot. Again, the purpose of this is to be talking about science research at an accessible level so that, anyone can understand. If you don't understand something, please do ask us questions.
Mm-hmm.
We really do encourage people to
even raise your hand in the middle of the presentation.
There you go.
Please do. Yeah.
Please interrupt us in the middle of it. Like, if you want to go up there, stand up like, hey, I don't understand something. Please do so. In terms of a few content warnings, we'll be talking about descriptions of medical procedures and terminology, as well as having some mentions of morbidity and mortality. Those are the more technical terms for just, for lack of a better term, just natural life and death processes. So take care of yourselves. If you need to step out for a moment, do whatever you need to. You won't offend us. You won't offend anyone. That's perfectly fine. A few disclaimers. We're not medical doctors, nor can we provide medical advice. And none of the research that we present here is our own. We have no conflicts of interest. This isn't our own work. We're not paid to put on this panel. We're only here because we want to. Mm-hmm. And most importantly, we're not representatives of PCF. Jody in the audience here is, but we are not, so we cannot speak officially or on behalf of PCF.
Yeah, any of the information that you'll be seeing about PCF in this presentation is publicly available information that you all could go ahead and go find yourself as well out there on PCF's website or any of the websites that are directly linked to within PCF. So, yeah.
Sounds good. Mm-hmm. Any questions before we get started? Sound good, everyone? Okay, looking good. So, first section, Cancer Biology 101, take it away.
Yeah, so I promise y'all, if you have not done any research on cancer or not had any family members with cancer, this will still be pretty easy to understand. We're just going to break down the basic idea because cancer as a whole sort of umbrella of an idea actually kind of means something. We have a lot of cancers that can go on in different parts of our body, but they all have a very similar root cause and root idea behind them. And the root idea is that cancer cells rapidly divide and multiply, and therefore they make more of themselves very, very, very quickly, and they break down the systems that would basically check that. So your body has natural systems to not let cells divide and multiply and grow all over the place all wild. But cancer cells find ways to evade that, and they find ways to spread themselves very, very quickly. And then once they do that, they can do all sorts of other things. But first, how do they do this? How do they divide super quickly, and how do they evade detection? Well, that's because of the DNA in our systems. And that DNA, of course, is kind of the blueprint for every cell that we have. It is the genetic material for every cell that we have. So cancer cells have mutations in that DNA. And what mutations just mean are changes. So those changes in the DNA, first and foremost, will make the cells start making themselves over and over again much, much faster, much more rapidly. And sort of that's the first step of it all, as you can see here on these slides. And then you can see here a nice little description of what exactly makes those cancer cells divide. So we call a gene that gets mutated such that a cancer cell divides and grows really, really fast an oncogene. So if you hear the term oncogene, that's what we're talking about. Something that has changed in the genetic code, a gene that has changed the genetic code that has caused the cell to start going haywire and making way, way more of itself. So all cancers need oncogenes. They all need those mutations to happen to be able to spread all the way throughout your body. So this is something that a lot of research goes ahead and targets, as you might naturally think. You know, what are these oncogenes? Let's figure out what they are, where they are, and how we could best potentially combat the mutation of these oncogenes. And then the other mechanism that every single cancer needs to become dangerous and to do the things it does is to evade cell death. So the body has a lot of mechanisms for which to detect that something is going wrong. It can even fix its own DNA. So a lot of the time an oncogene will mutate and then the body will actually fix that before it even becomes cancer. But if that doesn't work, then the body goes, this cell is no good and we can't even stop it from the inside. So let's just tell it to go ahead and make itself not live anymore, evade cell death. But then the cancer cells find ways to evade that system as well, and then they basically ignore the body's response to that. There are also other lines of defense like the immune system that then go ahead and also detect cancer cells, and the mission of the cancer cell then is to evade those responses. So it's basically like a progressive process, but the two most important things for a cancer cell to do is to grow really rapidly and avoid the methods of the body to stop them from doing that. And yeah, you might have heard of really impressive, important tumor suppressor genes. Actually, a really good example, BRCA1. This is a very common gene that we talk about in breast cancer. And this one is one where if you mutate it, then the cell is going to have less of an ability to stop itself from growing. The body's going to have less of an ability for it to stop itself. So that's why BRCA1, if it is mutated, often leads to breast cancers. So yeah, if you've heard that, well, there's a little bit more information on it. And then, of course, it's not just those two things. Cancer cells and cancer as a whole have a bunch of different hallmarks that make them so dangerous. They can make their own blood cells. They can evade the immune system. They can do all sorts of other things. They can make tumors, which are basically just big old clumps of themselves that do all sorts of scary things. But we're not going to be talking about all that today. Just know that there's a lot of properties of cancer that go beyond just the two things that we talked about. But all you really need to know, why cancer exists, grows fast, doesn't stop growing fast. It evades those the body's stop signs. It ignores them. Naughty, naughty cells. And even though cancer cells all have these sort of hallmarks, cancer is still really, really diverse. You can get cancer cells happening in every single part of your body, every single organ. There's some different spots as to where cancer is more prevalent to develop. Certain parts of the body you can see in this chart here will sometimes develop cancer more frequently. And of course, there are differences between the sort of sexes, although, of course, that's a very complicated conversation, and trans research is very understudied. But unfortunately, that's a little bit outside of the scope of our research, but I wanted to mention it as something that is very, very important in the field that we need to focus on more. But yeah, if you ever hear about the stages of cancer, this is also a very important thing to cancer research and to cancer treatment. The sort of lower stages, as it were, are often ones that are more localized. So if you're talking about stage 1 and 0 cancers, those are often ones that are in one place in the body, and those are more often surgery is often more successful with those kinds of cancers. And then when the cancer progresses, that's where it's going to start to go to different parts of the body, and that's what we call metastasis. And that's when it becomes a lot harder to treat because things like surgery, things like radiation therapy, those types of treatments get a lot more difficult when you're talking about lots of parts of the body instead of just one part. And of course, it also depends on what part you have cancer and how dangerous it is and how you're going to be able to tackle it. So even though cancer has a lot of hallmarks, which is what we call sort of those basic properties, there's still lots and lots of work to be done to tackle each different type of cancer individually because they're all super different. And that's why we need cancer research. Yeah, and on the sort of more clinical side of things, we're going to go over a little bit about how cancer is detected, and there's a lot of ways. It depends on how your cancer presents, how you're going to be able to find it, but the first step is often just a screening, a routine screening, or an appointment. This is why it's really important to go to your screenings, and of course, the Prevent Cancer Foundation is all about this, making sure that we catch cancer as early as possible because as we just went over, the earlier you catch cancer, the easier it is to treat. So those routine screenings or an appointment to discuss something concerning. So for example, lots of skin cancers get found that way. You find a little abnormality on your skin, you go to the doctor to talk about it. Or you're feeling symptoms. You're not feeling so well, you have pains, you have drowsiness, you have exhaustion, and from there, perhaps you find yourself at a diagnosis. But sort of in the later stages, a biopsy is when tissue is taken and examined for cancer signs, so this will happen a lot as well. Now, one of the projects that we're going to talk about later, a little spoiler, is finding some ways to do detection on a cellular level where biopsy is not required, and that is super cool because biopsy is a little bit invasive. But it is a really reliable way to tell whether cancer cells are present. And you can also even do like DNA screening, things in the lab that will sort of tell you exactly not only what kind of cancer that you have, but also sometimes even what genes are mutated such that you're getting cancer. And that can be really useful for genetic therapies and stuff like that, which we'll talk about a little bit more later as well. And for PCF, PCF has routine screenings for breast cancer, prostate cancer, lung cancer, cervical cancer, and colorectal cancers. And these are all really common types of cancers that have really diverse outcomes and are really prevalent in all of our population. So they're super important for being able to catch cancer early and is some of the most important work, of course, that PCF does. And then finally, what happens after you're diagnosed? Well, there are a lot of different types of treatments, and this isn't quite the scope, within the scope of our presentation to talk about super in depth, but all you really need to know is that there are a plethora of different ways that cancer can be treated, and a lot of cancer research is based around this as well, finding the most effective treatments that are not only actually effective in making sure cancer goes away, but also cheaper to implement and easier on the human body to take, because as I think all of you know, some forms of cancer therapy can be very, very intense and very difficult for the human body to take, and that can be very difficult. So a lot of current research is not only focused on what kills cancer, but what lets a human be happy, healthy, thriving while actually fighting cancer. And a lot of that is like immunotherapies that are really exciting, activating your own immune system to go against cancers, and a lot of genetic therapies, things that are looking at specifically the genes that are mutated within cancer and looking at that as the specific place to sort of attack, so to speak, with our research. And yeah, so that's kind of the general overview of what you need to know about cancer biology and cancer screening and treatment. So if you have any questions, now's a really good time for this section before we move on. Okay, fantastic.
Looking good.
I'll take that as we did well.
Awesome. So now here's the part where I step in with some more quote-unquote mundane section about how cancer research funding works. This is the paperwork and red tape, so to speak, that a lot of researchers are like, okay, I've got to apply for another grant, got to get some research money so I can actually do the work that I do. But this part is pretty important in terms of not only being able to obtain the funding that you need to do your work, but also just thoughtfully and clearly laying out how exactly you want your scientific experiment or your idea to proceed. So even the process of applying for funding or grants, this is an important section that basically all researchers and scientists have to be really careful about. Just write down exactly what experiments you'll do, how much you need, where each dollar goes, and what you hope to find at the end of that, your hypothesis, basically. So this is just a lot of structured. Please write it down, submit it to the appropriate grants agency, and just work through any of the comments or ideas that reviewers have. So. In general, how does this work? Like I said, you come up with a project, you write a grant proposal. That's the more proper term for it, is a grant proposal. You submit it to a funding organization. So my experience has been mostly here in the United States. If you want a research grant to do scientific research in the United States, particularly biomedical research, your main source will be the National Institutes of Health, or NIH. The NIH is the big government body here in the United States that's dedicated to funding scientific research using U.S. taxpayer dollars, and they coordinate a group of like 27 individual institutes underneath them, with the National Cancer Institute being one of the largest ones. So there's a lot of structure to this, but they manage a lot of research funding. So just being able to navigate this process is very important. A couple steps to actually get this grant is to go through peer review first. So once you submit a grant, an idea for what you want to do, you have to get it reviewed to make sure that what you're proposing is reasonable, that it can be done, and that it will have a meaningful impact of regardless of whatever you find or what you don't find, whether your hypothesis turns out to be correct or incorrect. Either one are both valuable pieces of information, so you really want to have it clearly structured when you submit your proposal. And then another part of that process is getting IRB approval, or Institutional Review Board. That's a group that's located within the various institutes in the NIH to make sure that you're passing safety regulations, that you're getting informed consent if you're working with humans, that you're following all proper safety procedures and laws in terms of handling hazardous materials. It's just a safety checklist, basically, is what IRB approval is. So assuming you get past all of that, and assuming you get an award, you can proceed as expected. You buy equipment, you hire personnel for the project if applicable, you complete your project, and then you write up papers and hopefully publish them at the various scientific journals that exist on the internet. When you get an experiment done and you have something you want to share, quite literally with the world, with the scientific community, you write it down in a scientific paper and then submit it to one of the many, many journals that exist across the world to be like, here's what I found. I wish to share this with others. Here's an introduction, methods, experiment, what we found, conclusions, all that stuff. And then at the very end of it, you make sure not only your papers are published, you send over reports to whoever funded you to tell them, here's how I made use of your money, and here's what we found in return. Just so the agency knows, hey, they went through with their experiment, and the money that they put towards it actually did something with it. So in a very broad sense, this is a general outline. I'll dive into some more specifics here, starting with where exactly this funding come from. Now, I mentioned this previously, but by far the largest funder of research grants is the National Institutes of Health. Not just here in the United States, but also globally. I think they're just the largest agency in the world that provides research funding. But it can also come from nonprofits and industry partners. Prevent Cancer Foundation is a non-governmental organization, so they're not completely affiliated with for example, the U.S. government or the National Institutes of Health, but they are as valid a source of funding as any other. So it just depends on what each NGO's or non-governmental organization's individual mission is for how they want to provide funding. Yep, I'm just going to step through this because I'd already talked about it. Some industries will also sponsor it if they feel that the company has a vested interest in a particular technology. So, for example, I'll throw out just a couple there. Not that I expect any of you to recognize it, but if you've heard of Illumina, that's a big biotech company that makes lots of DNA sequencing machines. They will often fund research on like experimental ways to make it easier to extract DNA from cells or make it easier to read the genetic code. And they have a vested interest in that because they want to build faster and better machines to be able to process DNA. So the research is technically sponsored, but of course the research itself is definitely like nonpartisan, non-biased. It has to be completely neutral in terms of what they found and what they didn't find. They can't over-exaggerate what they did. They can still accept funding from people who are are vested in a particular outcome of it, but they cannot be influenced by please show us this positive result. That's fraud, and that's not allowed. So you can do it, but there are lots of guidelines and regulations on how you use that funding, how much contact is able to go between the two groups in that case. And then finally, individual donations and sponsors are very, very, very important source of funding. So literally, quite literally, people like you who are sitting in the audience and donating to PCF are a very critical source of funding because as you've seen throughout this entire marathon, whether you donate $50, $10, $5, even $1, every single dollar goes towards a research grant that is put towards a specific purpose. If that dollar goes, if it's your taxpayer dollars here in the U.S., you can. There's a public list of grants that you can find on the NIH websites and all the various institutes as to how exactly that funding is being divvied up and where it's going. If you donate directly to an NGO like PCF, PCF publishes what projects they fund and exactly what projects they do, and we'll show those later on as well. So very important, all these different sources of funding. With that being said, what exactly are people or researchers and scientists supposed to put into a grant? Very broadly, there are two different buckets for this. There are direct costs, i.e. the money that goes directly to the project specifically. This can account for salaries of scientists, employees, staff, whoever may be that's working directly on the project. A lot of the times when you're hiring research associates or postdoctoral scholars or lab technicians or research assistants, their time has to specifically be broken down line by line as to how much time they're spending on each individual project. So, for example, I'll throw out for like a lab technician. A lab technician's role in the lab is to do all these different assays and do some biological, like cell cultures and whatnot. You're experienced with this, so you know a lot about that. But yeah, their funding has to be broken down line by line to say, okay, they're going to work 50% of the time on project A that deals with, I'm going to make something up, how to grow cells, how to grow cancer cells in the in vitro environment, and then 25% of the time is administrative work, and then another 25% of the time is working on another project with another collaborator. That is broken down line by line, and you explicitly write that into a grant. So it is very detailed in that aspect. Other sources of direct costs include equipment and supplies. Do you have to buy machines to do your research? Do you have to buy reagents and chemicals? Anything that you have to buy and put into a lab and physically manipulate, that would also fall under direct costs for equipment. And then travel publications are definitely a little bit lower on that scale, but those are funds that you can set aside so that you can specifically publicize what you find if you're able to publish anything and share what you found with the broader scientific communities you're involved in. The other big bucket is called indirect costs. These are costs that are not solely related to one individual project. So say you're running a research lab. You can work on a cancer project, but you might also be working on, I don't know, a project on another human disease. I work on sickle cell, so I can just say maybe you're also work on sickle cell part-time. If you buy something that involves both projects or can be used for both projects, that's an indirect cost because part of it is going towards the cancer project you propose, but part of it also is not. it's still a valid cost because part of it is going towards the thing you asked it to go towards, but you also have to account for that equipment can be shared. This is just a line by line item thing to make sure that every dollar is accounted for. So that includes facilities, the building you're working in, maintenance for any machines that you upkeep that can be used between projects, and administration for general administrative costs like support staff and usage of equipment, shared equipment, and rooms and whatnot. What exactly is funded? Lots of different things. I mentioned these previously, but you've got literal reagents, you've got server racks and computers in them if you do computational work. That big fancy machine in the top right corner is an Illumina sequencing machine. If you take a DNA sample from someone, if you take a cheek swab from someone, that's probably the easiest way you can get DNA from someone is taking a cotton swab, swabbing the inside of your cheek, and then just putting it in a tube and sending it off. That sample, that cheek swab, goes directly into that machine you see in the upper right corner, and then once it does so, you get a readout of all 3.2 billion base pairs of your DNA. It's actually pretty amazing how far DNA sequencing has gone. And then the bottom right, just a bunch of journal publication and magazines. Nature and Science are probably two of the biggest journals for scientific publications out there. That's, again, where you put your papers if you want to publicize them to the scientific community. So that is a small but essential part of the cost of a research grant. I won't belabor the point too much here, but because this gets really into the weeds, but this is really to ensure that every single dollar is accounted for and that every dollar is meaningful in the context of what you're asking money to go towards. In the United States, through NIH guidelines, there are several steps to this. The biggest step, and that everyone, from scientists to researchers to everyone else, knows and believes, are peer review committees. So this is where you get judged in front of a panel of your fellow scientists. So say if you're a cancer epidemiologist, your grant goes through a group of 20 other cancer epidemiologists that ideally have little connection to you personally, but are in a place or know enough about the subject to review your proposal in an unbiased manner. So that's saying, hey, I'm a fellow cancer researcher. I think this is a good idea and a good use of research funds. I am happy to write off on this grant. Or you could be like, no, I do not agree that this grant fits within the guidelines that we have for this institute, or I don't think this is a good use of the money that we have. I feel this project might be more efficient. It's really a back and forth between a committee of what is the most valuable use of the dollars and what will make the most scientific impact. So. And then the other step is advisory council review. So that's after it's passed peer review, you get reviewed by some of the folks higher up in the institute. It's just another administrative step to make sure, are the grants that the peer review has given us, are those in line with the institute's mission as a whole? So for example, for the National Cancer Institute, for these grants that we've gotten, is it in line with the National Cancer Institute's mission of helping to find the causes of cancer and to find better cancer screenings and to prevent cancer in the first place? So that's like a more broad conceptual checkpoint. And then one big point for NGOs like PCF, the good thing about NGOs actually is that, again, they're a perfectly valid source of research funding. And the benefit with those groups is that they give researchers more opportunities for concepts. And that concept word is pretty important. That means more exploratory things. So something that might be too experimental for the NIH, for example, can be funded through a mechanism like PCF, because maybe you have like a very out there technology that has never been tested before for maybe detecting colon cancer like 20 years earlier than we're able to. That might be too risky for the NIH to invest money into because it might not generate any positive results or whatnot. But for NGOs like PCF, they can fund whatever they want. So it gives you a lot of flexibility if, for example, you want to try out some things. So each organization obviously has their own mission and what they want to fund, but it's if you look around a bit, you can find organizations with different missions and exactly what they're interested in funding. So it's a very wide world out there in terms of what folks are willing to fund. All right, I've yapped enough a bit, so if you want to talk about the projects.
Yeah, and we're going to give y'all a little overview of how PCF works in terms of the research projects that they fund, and a couple of examples of projects that are currently being funded by the Prevent Cancer Foundation, so you can know where some of those donations are going right on towards. Besides, of course, all the fun stuff that GDQ puts on. So, how do they support cancer research? And just as Curry was talking about, PCF themselves provides research grants and fellowships to researchers. So you are able to apply for funding from PCF in particular directly, and then they will review your application and decide whether they'd like to fund you. So it's a very similar process in terms of that sort of broad conceptual way to something like the NIH. Of course, there will be structural differences, but it's the same general idea. Ask for funding because you think you have something cool that might end up contributing towards science as a whole. And of course, for the Prevent Cancer Foundation, the vast majority of the funding that they use goes towards cancer projects in specific. So if you have a really interesting cancer project, and especially ones that are related to prevention, PCF funds a lot of those. You can ask PCF, hey, would you like to fund my project? And something that's actually really, really cool about PCF and something I love about their website is that they have a research award database, and it is really comprehensive. It's searchable. So if you want to see all the things that they fund, and if you want to search by different topic, different type of cancer, all sorts of stuff, you can go right towards https //preventcancer.org/research in order to find what you'd like to find. It's really, really cool. And also this page has a lot of general information on their research practices and a lot of the projects that they are currently funding that they are sort of featuring. So this is a really fun page to go to, really interesting page to find out all the information you need. So if you're asking yourself many big questions after this about PCF, this is a really great place to start. Just wanted to give a little information on sort of PCF's general impact in terms of research funding. So they have a 10-year research grant and fellowship impact fund, and between 2014 and 2023, $8.1 million in research funding was issued to support 83 grants and fellowships. That is really, really impactful stuff. Those are 83 projects that would not have gotten funded if it weren't for PCF existing and giving their funds towards those projects. And it takes a lot of money and work to do a single cancer project. That is a lot of impact that PCF is having directly on scientists around the United States. And 59 institutions were represented in this funding, so they work with a lot of different institutions in terms of who gets that funding as well. It's very diverse. And then they also have returns from this investment listed. Now, of course, the overall mission of science is to better humanity and to better our knowledge, but there's also the financial aspects to things. We always have to make sure that we're running well, and PCF is very financially responsible. They have a very high rating in terms of how well they do, in terms of where they spend their funding and at what they are able to get back from it, and how responsible they are with their funding. So this is some hard proof of that concept, which I think is really important to keep in mind when you're deciding what kinds of charities to support. PCF is really responsible with what they do. And here's a really cool example project. We're going to talk about two that are listed on their website. There are tons more like this that you can just go read about. They have really cool little bios, and you can see the actual websites for these people there. It's all wonderful. But our first one, Tamara Berger, MD, MHA, is doing some really cool non-invasive methods to detect melanoma research. So invasiveness is a huge thing with cancer screening, because the more invasive a method is, the more risks it has, and also, possibly even more importantly, the more people don't want to do it. It's really important for a screening to be easy and accessible for people to want to go ahead and go for it. So this research is super important because of that. Tamara is developing some non-invasive methods to detect melanomas. And melanomas, you might have heard that word before, they are a really dangerous form of skin cancer. About 80% of mortality from skin cancer, so 80% of deaths from skin cancer are specifically melanomas, even though they make only 5% of skin cancer diagnoses. So that is really, really dangerous. It's super important to know if you have a melanoma as fast as possible. So having these non-invasive methods be developed is super important in being able to catch these really dangerous cancers early and be able to treat them. So one kind of method they're working on is some adhesive tape. So you could stick that onto your skin, you remove it, and then that collects the cells. And I mean, that sounds a lot better than having a doctor sort of scoop out part of your skin, right? Sorry for the sort of visceral description, but doesn't that sound easier? So having these sorts of methods be developed is super important in order to get more people better health outcomes sooner. And then another example project that they fund, this is Jim Hu. He's an MD and MPH, and he researches prostate cancer messaging on the internet and seeks to improve it through a variety of methods. So this is less of a sort of direct in that wet lab scenario, which is where you're working directly with cells and making stuff up. This is more about the public messaging that's going out to people, which is equally important. Their lab is looking at how do we get the information about prostate cancer and prostate cancer screening to the people that need that information. And how reliable is that information on the internet? How accessible is it on the internet? And how can we sort of assess it for these metrics and improve the ways that we're talking to people about prostate cancer. They're also surveying people about their attitudes towards prostate cancer. So that's really important to survey the population on how they feel about a lot of these things, because as we just said before, the ways people feel about cancer screening is often going to determine whether they get a cancer screening. So if people are not a big fan of a screening method, or if they don't trust that a screening method works, or et cetera, they're not going to go get it. So we really need that information as to how people feel about different screening methods and what they want to sort of see in their health, in their world of their health. So those are just two examples of the types of projects the PCF funds. They're a little different from each other, but they both are under this sort of broad umbrella of cancer prevention, early detection, and making sure that we're sort of getting the best outcomes for public health. Do you have a question?
Yep.
Go ahead.
The question might actually be a little bit more directed to your guest.
Oh, yes.
But it's one of those curiosity things of, we think of cancer research, as you're talking about it, we're studying human genomics, things like that. At the beginning of this event, I found out my cat has cancer.
Oh.
So it's one of those questions of, obviously the vast majority of this research is focused on human cancer research. How much of that is also applicable to other animals? Because as much as we like to focus on ourselves, humans are just a small portion of the animals on this planet.
That's such a good question. Yeah, I'll let Jody get the first crack at that.
Yeah.
Yeah, some information, yeah.
I'm sorry. I'm sorry about your cat. I mean, is this working? Yeah.
Yeah, it
Yeah,
works.
it is, it is.
It's a little awkward because my back is to you. I can just yell. I'm going to yell. Okay. Very often for us, it's the other direction. So what we're seeing in prevention and screening is that so there's zebrafish is an example of where there are learnings because they match the human DNA. So there's structures of fish, for example, that are can be used to test different models for us in a humane way. And where there is uptake in veterinary medicine in particular, is less on the treatment side and more on the detection side as well. So it's come a long way, though. There are oncologists within the veterinary space that are happening, but for us, the translation is taking much longer than it should be. But in many places, we're behind animal medicine. So when my family are all on the veterinary side, when I say, okay, there's a great new thing that's happening in cancer, they're like, we've been doing that in horses for 20 years. So I'm happy to take your question back and ask my family if there's anything there. But it's not happening as much as it should, probably.
Yeah, good answer. Yeah, so every organization has a different focus. An organization like PCF may have a different focus than another organization, and a lot of that directs where their funding goes to. So it depends on what organization you're looking at as to specifically what they're funding in terms of veterinary medicine versus non-veterinary medicine. But as Jody pointed out, a lot of the time it goes in the other direction. A lot of studies in veterinary medicine actually inform human medicine in a shockingly impactful way. It's really, really cool and interesting. This is one of these things that I feel like we should talk more about But the advances in veterinary medicine can happen so much faster than in human research because of the structures around human research being so strict, which of course they need to be for ethical reasons. But there's a lot of things that in veterinary medicine you can find out results a lot more quickly. So we take a lot of findings from veterinary medicine, go, oh, would this be applicable to human medicine? And we look at that. So that's just a cool and interesting thing. And also, evolution is fun. And that means that a lot of the things that humans have going on, a lot of our other related animals, so like mammals in particular, there's a lot of crossover actually in how the mechanics of human cancer works and the mechanics of mammalian cancers works. So there's a lot we can learn from each other and a lot of crossover in that respect.
Yeah, I mean, I could just sit here and nerd out about cancer biology and genetics all day, but yeah, it's just you know, I like to think of the fact that yeah, bananas share 50% of your DNA with humans, and we're like, huh, what? That is both true and not true at the same time, because obviously bananas are not humans. They don't have hair or eyes or arms or anything, but they're made of cells, and humans are made of cells too. There's a lot of things that you can't see physically that we share in common with every other living thing. We're both made out of cells. We need to have checkpoints and regulators of cell growth, division, and death. There's lots of shared commonalities even between two very distinct organisms. So that's why we're able to translate discoveries that we find in other mammals or other primates to humans, the other way around as well. So it's very, very interesting. Yeah.
Yeah, absolutely, absolutely. And then, finally, we're going to talk about some of the stuff that's happening in the world right now.
You got it. Yeah. This is also part of me just nerding out about all this. So, all these, I'm going to show you 3 different publications here, and I'll just say real briefly that these have all happened within the past few months. So this one has been published in October, late October, October 29th. I got these from a friend who shared these to me from, I think, a science communicator called Dr. Cal. I believe that was their name. But yeah, this is a paper showing for evidence for improved DNA repair in a long-lived bowhead whale. We're taking something that we learned from a whale, and we're translating it into a human genetic context. We're saying we've noticed that DNA is being repaired, or rather just not being destroyed as much in whales, and we're trying to figure out how that can apply in a human context. Can we use the DNA repair mechanisms in a whale and possibly come up with something to help promote cancer screenings or cancer treatment? Of course, it may sound far-fetched, but this is really where scientific discoveries and progress starts. I mean, if you take it on at face value, yeah, what are how much similarity does a whale have to a human? It feels far-fetched, but this
so
is
much.
really there's a lot, right? But it's really a step-by-step process. This is an iterative process, the process of scientific discovery and research. We have found here that whales can maintain genome integrity, so that means their DNA doesn't mutate as much. And as we learned from earlier, if your DNA mutates much more often or in certain ways, that's how cancer starts. So maybe we can take a lesson from the whales and be like, let's see what we can learn from the whales of what they're doing differently from humans, and seeing if we can use it somehow to prevent cancer in other organisms. So this is at the very start of the pipeline. It's very new. It's very interesting. This should have been an animation, but that's fine. That's fine. Again, this one published last month, I think exactly a month ago. This basically talks about a new technology about how we can use lab-modified T cells. So T cells are a form of immune cell you have. They help keep you healthy. We have learned of a way we have come up with a way to edit these, what we call CAR7 T cells, to help patients with leukemia. So these are people that already have leukemia, but we can help them, but these cells help them get better at a much quicker and better pace with less side effects than most of the other cancer treatments, like the typical ones like chemotherapy or bone marrow transplants that we've seen before. So this is basically, we take T cells, we edit their own genomes in a particular way. So it's not like a whole living person. It's literally just a very microscopic immune cell that you cannot see. We edit that genome, we put it back into the person with leukemia, and it helps them get better rapidly. It's amazing.
So cool.
It's so cool, and it also helps because after you do this kind of leukemic remission, as we call it here, you can also go ahead and do some hematopoietic stem cell transplantation. I promise I can say these things out loud. In a nutshell, that means replacing blood cells in the patient's body, because if you go through chemotherapy, that wipes up your immune system. You have to get new bone marrow put in, or new stem cells put in. Stem cells are the cells that allow your body to make more and new cells out of those stem cells. Stem cells are the core, and then from them is made the diversity of all the other cells in your body. So it's really cool. And then the last one here was published literally 2 days ago. I looked this up 2 days ago just because of like, what's new in cancer research? Cancer research is probably one of the most active areas of research in the biomedical world. So this one I found to be pretty interesting. This is basically making organoids. Organoids, if you think of organs as like your heart, your liver, lungs, stomach, etc. Organoids are very, very, very small versions of that. So instead of talking like a macroscopic heart or something, literally a pile of 100 cells on a cell culture dish you can't really see. That is what an organoid is. And
Our lab worked with organoids.
oh,
when
perfect.
I
I feel like you could talk about this. I'll mention this real quick and I'll let you chime in.
No,
but,
they're just so cool.
It's really
This is
cool.
one of the major breaking things that is in science right now. Just to kind of give you perspective as to where we're going with science, organoids are going to let us be able to test a lot of basically treatments, test a lot of other things in a way that's way less invasive, way less expensive, way less difficult than doing something in like a human body for a lot of, especially those early stages of that treatment testing. And that is just critical. It's going to make research so much more efficient and so much more accurate. It'll be great.
yeah, I mean, it's organoids are really cool. Beyond that, this particular organoids that they found that they were able to work on in this specific paper is that these are patient-derived. So again, you take like a cheek swab of your own cells, like literally non-invasive. You just take a cotton swab, take some of your own cells. You can grow an organoid from your own cells. And the benefit of that—
It sounds like science fiction, but it's real and it's happening.
it's actually real. It's actually happening right now. We are working on this right now. And the benefit of that is that these are your own cells. It's not someone else's cells or like a cell line that may not be compatible with you. If you grow an organoid from your own cells, you have a very high confidence level that whatever you find with this organoid from your own cells will be compatible with you personally. This gets into the idea of like personalized cancer screenings and personalized cancer treatments. So instead of just doing a broad spectrum like chemotherapy that just blasts radiation at your cells in hopes that the cancer cells die, you can specifically target just your cancer cells, and you don't need to like throw the kitchen sink at everything. That is one of the potentials of developing these human organoids, especially using your own cells. Just amazing stuff.
Yeah, we're just nerding out now.
Yeah, this is nerding out.
So if y'all have any questions about anything we've talked about, from the way that cancer research funding works, how cancer works in general, what PCF does, and of course we have our lovely CEO here, so she can answer specific questions really well about PCF, or what's going on right now. Do let us know either now or at the end of the presentation.
Yeah. So I just want to end this with a few concluding points. So what's the final message? We will get there. I fully believe in the cancer research we're doing right now, and I believe we will make tremendous discoveries. It's really been an exponential growth of what we know about cancer. Even within the past 20 years, actually, within the past 20 years ago, it was only 2003 that we first sequenced the whole human genome. That you might have heard of it. It's called the
Most
Human
of us are
Genome
older
Project.
than that.
Most of us are older than that. So it happened within our lifetimes. But ever since we were able to sequence the whole human genome, we've learned so much more beyond that. And it happened while we were all still alive. So we're still in the very beginning discovery phase of how much more there is to know and learn about this. So I believe we'll get there. Again, like I said, been saying, new research is coming out every single day. I've showed you articles from 2 days ago. It's a very active field. Truly exciting if you didn't get that already. And we're seeing the results of cancer prevention in real time. So if I flip to a few different slides here, actually give me 1 second. Yeah, I'll go to this slide first. This is a graph taken from the cancer epidemiology website that the NIH runs. What you're seeing here is a graph of rate of new cases as well as a death rate due to cancer. The rate of new cases is, it's been rising gradually. It's probably remained a little bit more constant nowadays, but that can be interpreted a lot of different ways. And one of the ways you can interpret that is that we are lowering just basically the incidence of cancer as we learn more. Here's how you prevent cancer. Here's how you prevent here's how you prevent skin cancer. Make sure you wear sunscreen. Make sure you obviously get some sunlight exposure, but not too much. There's lots of different ways or lots
Regulations
of different ways
in the food industry and in
Exactly.
environmentalism are, a huge impactor on how many people get cancer. And so we're not only preventing cancer, but as you were, I think, about to say, we're detecting
we're
cancer
detecting
more.
it. Yeah, we're detecting it more and preventing it more because there are certain stages of cancer where you can detect it, but it's not cancer. It's like almost cancer.
If you hear the phrase precancerous, that's kind of what that's
what
referring to.
Exactly.
Yeah.
Yep.
It will be cancer if we leave it unchecked, but it's not yet.
It's not yet. And because of all these different screenings, advanced screenings and prevention methods, that death rate right there is going down. You can clearly see that. The death rate from cancer is going down, at least here in the United States. So we're making progress here. And this is the 5-year relative survival rate of cancer. So say a person does develop cancer, how far out after 5 years are you still alive, basically? That line is going up.
You can see in just the 70s, 50% versus now over 70, around 75. That is such a massive difference in how many people are still with us 5 years after they first got diagnosed in the current year. Just incredible impact.
You can tangibly see it. It's amazing. We're actually making real ground on this. So we are hoping to not only cure more cancers within the next couple decades, but also find prevention methodologies for a lot of them as well. That is the hope, to both cure but also prevent cancer.
And
So-
for PCF in particular, the earlier you get diagnosed with cancer, the better survival rate you're going to have, the better health outcomes you're going to have. So that's also a result of us detecting cancer earlier and not, finding out, right before a person is about to die or even after they've died, why they died was because of cancer. Finding out instead in the stages, 0 or 1, you have cancer, but there are options, and we can help treat it earlier is leading to these better survival rates as well.
So I'll go back a couple slides, but really the message is
You,
yeah. Your donations are directly going towards these cool discoveries and helping prevent cancer in the first place. Coming up with all these new ideas, coming up with these new technologies, and just helping save lives. It is a literal, tangible impact that all of your donations have and where all this research funding goes towards. So thank you to everyone who's here. That is it for our presentation. So we have a bunch of references, but that is it from us. So we'll take any questions if you have them. Thank you so much.
yes. And feel free to come up to
Please
the mic.
come up, yes.
So that everybody can hear you.
Yes.
Yep. Make a little line. Yep. You're
Nah,
good.
there's no shame here. It's all good.
Earlier, when you were talking about grants, at the very top of the slide, it said generate the project instead. So, or just generate the idea for the project. My question
Yeah.
was, how much, what percentage breakdown I would say of projects are sort of generated from the researchers themselves being like, oh, this is a project I'm thinking I want to do versus maybe a more administrative side saying, hey, we have an idea for a project. What would you do? And I guess also for the PCF aspect, how hands-on are y'all? Do you have your own ideas that you approach researchers with, or are you more so just when people approach you, that's when you get involved?
Fabulous question, by the way. That is a great question. Yeah, Jody, definitely go through that.
Jody will come on stage.
Yeah, this Curry has a little bit more experience as a very active participant in the field on this one.
Yeah. So, just to make sure I understand your question correctly, so this would be how much time you spend or rather, how much of the project is more direct research versus just administrative parts of it? Is that your question?
The original idea of a project almost. It's
How,
like
how much is top down?
Yeah.
your idea versus like an administrator telling you to do.
I see. Okay, yeah. So the question is how much of the project idea would be your own versus someone from an administrative side telling you what an idea should be. Yeah. So I will tell you right now that as someone who's written grants myself and gotten some funded, this idea starts with you. You are the ideator. You are the person who writes up exactly what you want to happen. Obviously, you'll have a lot of peer input. So, for example, if you want to collaborate with other labs because you have particular things that another lab is an expert on and you could use their expertise, obviously you'll get input from that. But as you're writing the grant, that is 100% your idea. And then whoever you send it off to or whoever administrator needs to check off on it, I mean, they can choose to, approve or deny it, but they can't really do much in the way of do anything beyond suggesting any changes to your grant. And you can accept it or not, but the idea is fundamentally yours to write because it is your grant. You're in charge of executing the proposal. So above all else, you need to be comfortable with what you're putting in there, and you have the power to dictate what you want your group to do.
Yeah, so ultimately, scientists are actually creatives. And
Yeah.
a lot of people don't really think about it this way or don't know that that's the case, but you actually have to have a really strong creative muscle to be a good scientist because you have to be able to look out there into the world and think of novel ideas for projects, novel ideas for what to do. So it's really all about the researcher's vision and what they know and are able to do. Because as Curry said, you're the one executing that project. So the best person to write up a grant and to have that idea is the person who is going to do that project. So that's fundamentally how our science is sort of structured, at least certainly in the United States, but also mostly globally, is very bottom up. It's also a conflict of interest a little bit if the administration has things that they want to see out of researchers. So this is something that most of the time is avoided in terms of suggestion. Yes, things can be approved or denied, but oftentimes where those ideas are going to come from, and more than just oftentimes, almost 100% of the time, is from the researcher themselves. Yeah.
That's a great question because it's
Really good question.
It's in terms of the Prevent Cancer Foundation and how we think about influencing research. While we wouldn't get super specific. So generally, the researchers are telling us, the science community is telling us what's happening from the research space, but having more targeted calls for research has been helpful occasionally. So 5 years ago, we said we wanted to align with the World Health Organization and the elimination of cervical cancer. So we did a call for all cervical cancer prevention and early detection related grants. We're facing a little bit of a different challenge as we think about 2026 because last year, wanting to be very inclusive in our grant cycle process, because we cover so many cancer types, we get an enormous range. But what's happening now, as the federal landscape has changed and the funding environment has changed, we saw almost a 400% increase in our research grant applications. And it's cumbersome because you have to get two experts in each area that the research is coming in and to review that grant. So you're talking about hundreds of people sometimes. So we're going to have to look at how we refine and what call we do for 2026 to make it really specific. So sometimes strategically we do make more refined calls.
That's a really good thing to know. I think is really cool to know how you all sort of find your grants and what you're looking for in researchers. Also, just another thing that y'all are helping with here at GDQ with the landscape changing, with more demands for help. What we're doing here is really important. Do we have
Very
another question? Sorry.
Very.
Oh,
Okay.
well, there you go. Yes, go for it.
Since there's so many different root causes and types of cancer, is the research kind of perspective that every type of cancer needs to be addressed individually, or is there a significant amount of overlap between, say, research that's done on lung cancer affecting concurrent research going on with, skin cancer or breast
Great
cancer?
question.
I mean, is there a lot of,
Yeah,
I
another
guess,
great question.
efficacy across the board for those different types of cancer research and treatments?
That's a great question. I'll look at Curry first because that's the genomic. That's
Yeah.
that genomic
Yeah,
piece
yeah.
to some.
And let's make sure to repeat the question just so the recording gets it.
Absolutely, yeah. So the question was, how much does research on one particular type of cancer inform research in another type of cancer? How translatable it is, basically. That's the term we like to use. How translatable it is. I'll say that it's both at the same time. It's some of it is translatable, some of it is not. I'll go back to the point I mentioned previously of there are lots of things that lots of cancers have in common. They share the same common hallmarks that Satan talked about of all cancers go through angiogenesis, so they have to make new blood vessels for themselves. They undergo metastasis, so that's splitting off cancer cells to migrate somewhere else in the human body and make new tumors. So there are things that are common to all cancers, but beyond those like shared mechanisms, a lot of it does look differently. A lung cancer looks different from a skin cancer just because skin cancer is one that is very much affected by like the amount of radiation that comes onto your skin, and lung cancer is affected by like how many particulates in the air you inhale, and whether those particulates do any kind of significant lung damage. The way these cancers form are very different, but once they form, they tend to act in the same bodily pathways, so to speak. So there's a lot of differences, a lot of similarities at the same time. It's kind of a mixture of both.
And I can add to that and saying where it's being impacted is in early detection or cancer detection, because those areas where the tumor types or the body part types of cancer are acting similarly, in many cases, it's called circulating tumor DNA. So cancer sheds DNA, basically. And so what's happening is these blood-based cancer detection tools are being created that says, okay, there's dozens of cancers that are shedding once it's developing in your body, that's shedding DNA. We can do one blood test and be able to pick up a signal of those different cancers. So it's influencing very much on the detection side.
Yeah, that makes sense. Yeah, and basically the reason why they'd be fundamentally different, as Curry kind of touched upon, is the genetics. So every cell actually looks pretty different in your body, depending on where it is. And not only does it look different, like if you literally looked at it, it would look different, but the DNA is different. So the different genes that are going to be mutated that would cause the cancer in a skin cell versus like a lung cell are actually going to be different. So that's sort of driving where the research is different. But as you said, you can make a screening method that might detect for multiple cancers at once because it looks for those different hallmarks together, or it looks for a gene pool together. So yes and no.
Yeah.
Are there any more questions from the audience? Fantastic. Thank you all so much.
Thank you so much.
You guys, that was great.
Yay, thank you.
Thank you to all
Thank
of
you,
from-
everyone.
From us.
Thank you.
Well
All right.
Thank you so much.